All right. Thank you so much for the introduction, and thank you for ONT for having me here to speak today. I just want to start by saying I've learned a lot from this community and a lot from this meeting over the last few years, so this is a really exciting opportunity for me to be here and to talk a little bit about my project. I have nothing to disclose, but I am interested in talking to you about how I'm using ONT to comprehensively characterize tandem repeat expansions in the human genome. So just a little introduction into what tandem repeats are and why they're so important to study in human disease. Tandem repeats are a type of structural variant where a motif or sequence of DNA is repeated adjacent to one another in the genome. And these exist naturally in our genomes, but sometimes they can actually expand beyond pathogenic lengths and cause disease. And as of now, there are over 60 monogenic disorders that are associated with tandem repeat expansions, but there are more complex disorders that have been discovered in recent years, and I'm sure will continue to be discovered over time. The reason it's so important to comprehensively characterize these is because longer tandem repeat expansions often lead to more severe disease and earlier age of onset. Now, for most people, this is where characterization ends, where they'll mostly only look at size of these repeats. But there are other factors that are quite important to get a holistic picture of how these repeats contribute to disease, including methylation and sequence interruptions within the actual tandem repeat. So that's what I'll be talking about today. Now, just before I get into the workflow, the reason it's so important for us to be using sequencing technology like ONT is because molecular methods like southern blots and repeat prime PCRs, which were typically used for diagnostics and are said to be the previous gold standards, take forever. That's what I used when I first started my PhD, and I was very, very lucky to start integrating ONT sequencing into my workflows to speed up this process and, again, characterize these much more comprehensively. So just getting into our workflow, once we sequence DNA with our sort of workflow of choice, whether it's Cas9 sequencing, adaptive sampling, or whole genome sequencing. We then base call it, and we've done it on both Guppy and Dorado, and then align it to a reference genome of choice. So the input to this workflow is the reference genome of choice, a list of repeat loci that we're interested in looking at, and the actual BAM file that we've aligned to the reference genome. From there, what we do is we actually construct a repeat genome graph where every single node or every single repeat that we've introduced in the configuration file becomes a node that loops into itself, and we can specify multiple motifs. Simultaneously, we're extracting reads of interest, which overlap the actual coordinates that we've inputted in our config file. From there, we use minigraph to actually align the sequence to the graph, and then use custom scripts to parse those reads and output both plots and quantification of repeat genotypes. What we also do is we integrate methylation into this. So we actually use existing workflows from Epitome to use modkit to call those methyl tags, and we use custom scripts to actually assign methylation within the repeat. Now, what we're doing that's a little bit new is most people will usually stop at looking upstream and downstream of the actual repeat, but won't look within the repeat itself, because by default, these tools don't output that information. So we're just introducing some custom scripts to do that. The final output of this whole workflow is phased tandem repeat genotypes with confidence intervals, average methylation per allele, and also repeat distributions and methylation plots that integrate all of those things. Now, just to get into a little bit of genotyping and benchmarking to show you how well this tool is working in our hands. So I molecularly validated repeats in both plasmids and in cell lines using southern blots and repeat prime PCRs, which did take a while, but that's what we do. And on the top graph, you'll actually see the plasmids, and in the bottom graph, you'll see Coriell cell lines with various different repeat expansions. And on the x-axis, you'll see the molecularly validated sizes, and the y-axis is the STRcount or STRcount estimates. What you'll notice is that irrespective of the actual repeat that we're looking at, STRcount is quite accurate and is positively correlated with molecular sizing in all of our samples. We were quite happy with that, so we actually looked at how this changes across different base calling configurations, because sequence accuracy is also a little bit important to us. Now, I'm actually showing an interesting example of GC-rich repeat. This is a repeat in the FMR1 gene, which causes Fragile X syndrome. And what you'll notice is going from top to bottom, fast, high accuracy, and super accuracy sequencing. It's the same 7 reads, and the canonical CGG repeat is in these blue bars with other motifs represented by different colors. You'll notice that the sequence is best resolved at super accuracy sequencing. But on the right, I carried out an experiment where I actually downsampled a lot of these different repeats, including the CGG repeat, and looked at it at 5 reads in each of these different base calling accuracies. And we found that STRcount was accurate even in fast base calling methods, up down to 5 reads. So just another set of validation. We actually also orthogonally validated on the 8G002 genome in a bottle truth set with PacBio data using a tool called Target that was developed for PacBio. And what we can see is both in the smaller and larger alleles, we looked at over 170,000 loci, filtered down to just over 160,000 loci, and we see very good correlation with the PacBio data as well. In fact, we actually found some larger repeats in the ONT data set that we weren't able to see in the PacBio data set. And this is just showing you a match rate by tolerance. So if we increase the if we looked at perfect matches between ONT and PacBio data, we see about just over 93% of match. However, over 99.5% of our calls were matched if we actually account for ±3 repeat units. So we're falling within a very good range of accuracy. Now, just to talk a little bit about methylation and repeat instability. So repeat instability is a phenomenon where in somatic tissue, the repeat actually expands more than the germline tissue, and this can affect disease outcomes. Over here, I'm again showing an example of an FMR1 CGG repeat. This cell line in this patient actually has 477 repeats reported with full methylation. This is a long-range PCR in the plot on the left, which actually shows a smear indicating that it's not just one single repeat size, but several. And on the right, on the top, I'm showing the STRcount quantification plot, where you can actually see that distribution really nicely and quantify it. And this is the methylation plot, both upstream, within, and downstream of the repeat, which shows that the repeat itself is generally methylated. This is a similar example in C9orf72, where a G4C2 repeat expansion causes amyotrophic lateral sclerosis, or ALS. And in this case, the patient was reported to have 770 repeats without any methylation. Again, the southern blot on this slide actually shows that there's a primary band at 770 repeats with multiple faint bands, which might indicate somatic instability. You can actually see that quantified in the plot on the right. But what's really interesting about this sample is the difference in methylation. Now, on the top, you'll see in blue the actually unexpanded allele, where there's no methylation at all. But in the other plot on the bottom, you'll see the expanded allele, where the expansions that are shorter, so around 200 repeats, are unmethylated, but the longer expansions are fully methylated. And that really highlights the complexity between repeat instability, size, and methylation, and allows us to get a better picture for both prognostic, diagnostics, and looking at why our patients have certain outcomes. Now, I'm just going to finish off with a quick case study as well. We were actually looking in a cohort of patients to understand whether tandem repeat expansions contribute to schizophrenia. And in this case, I'm looking at the specific gene called PTPRN2 in a proband who has schizophrenia, where variants in this gene and differential methylation specifically are actually identified with schizophrenia. So in this gene and in this particular repeat, the reference sequence is a CCT-GGGT sequence. But what's really interesting is our proband actually has several sets of alternative alleles. The reason that's interesting is because the alternative alleles, which you can actually see separated on this plot, actually open up more methylable sites. So then we looked at whether there was differential methylation in these different haplotypes that could be contributing to this disorder. And sure enough, again, this plot is separated by haplotypes, and in the middle, you'll see the within-repeat methylation. And you can see that the top allele that has all those alternative sequences actually do have more methylation. And we confirmed through trio sequencing that they inherited this from their father, but the repeat expansion itself was much more expanded in the proband, and therefore much more methylated. That's the end of the case study, and I'll quickly summarize by just saying the reason we developed STRcount or STRcount is so that people are able to actually look at multiple different factors such as size, instability, methylation, and sequence holistically, because that really, really paints a much bigger picture and a better picture of why these tandem repeats contribute to disease. And we've learned that repeats are quite complex, but analyzing them doesn't have to be. So with that, I'll end by acknowledging all the members of my lab and our collaborators for making this project possible. Thank you so much for listening.